The Gnatwork is basically a 3-year GCRF grant which is looking at building a network of researchers on sandflies, blackflies and biting midges. So we're now 2 years into the grant. It's a 3-year grant in total, so I'll just be updating you on the progress to date. So a little bit of background into the Gnatwork to start. It was born out of a GCRF grant, which was to create a number of interdisciplinary networks around challenges relating to vector-borne diseases. So the Gnatwork is one of 4, and it started in June 2017, run by Dr. Simon Carpenter and Professor Mary Cameron at the London School of Hygiene and Tropical Medicine. And it's looking at creating a network around neglected vectors. So blackflies, sandflies and biting midges all share similar technical difficulties. They're all really, really small, so they have wing lengths of less than sort of 2 millimeters, making them very difficult to work on both in the lab and the field. These 3 vector groups have also had a number of fluctuations in funding throughout, for quite a long time, meaning it's been really difficult to maintain a critical mass of researchers across the 3 groups. Therefore, it's the aim of the Gnatwork to basically join researchers on these 3 groups, get them to work together, create techniques that can be used across the 3 vector groups, and therefore create a more resilient research base. So the Gnatwork's doing this through 2 main ways. Firstly, through funding pump prime studies to connect researchers, validate new techniques and generate novel data. And then secondly, through hosting annual workshops which will teach early careers transferable skills across the 3. However, today I'm just going to focus on the studies that we funded to date, basically let you know what we've got up and running at the minute and tell you about their aims and what they're working on. So we're running a total of 3 pump prime funding calls across the duration of the grant. We've got a total of £600,000 budget for this, and our first call was run in 2018 and it was named Validation. So this call basically wanted projects that validated new techniques that could be used multi-vector group or transferred techniques from one vector group to another. And we were really looking for projects in this call which had a high likelihood of success, so were likely to generate data. And we funded 3 projects in total, and they all started either at the end of last year or the beginning of this year. So the first project we funded is run by Matt Rogers at the London School of Hygiene and Tropical Medicine, and it's used to see if FDA cards can be used to monitor infectiousness. So this is into now its final 3 months, but it's based on the premise that when a vector feeds, it basically expectorates some of the pathogen onto the food source, and that can then be detected from the food source. So the idea is to sugarcoat FDA cards. The vectors will bite the FDA cards, take a meal, and leave some of the pathogen behind, which can then be easily detected through molecular methods. So this has already been done for mosquitoes, so for arboviruses, but he's looking at doing it in sandflies and midges to see if he can detect leishmania. And the fieldwork for this will be done in Ghana and Ethiopia, but the idea is eventually to move these FDA cards into like CDC light traps to basically increase the amount of data you get from your trap. Our second project that we funded is now about halfway through, and it's run by Professor John Colborne at the University of Birmingham, and it's looking if nanopore sequencing technology can be used for real-time surveillance of vector parasite populations. So what John basically wants to do is take large pools of adults and see if he can mass screen them using this technology, both for the parasite load and to identify the vector species. And this will be done by looking at mitochondrial DNA ratios. All his work is going to be linked to researchers in Ghana, and the basic idea behind this is that it's an easy way to get sequencing data in low resource settings. And although he's focused on blackflies and looking at Onchocerca, which is the parasite for river blindness, the technique is actually applicable to all vector groups. And the final study we funded in our validation call is actually being run by Chris Sanders here at the Pirbright Institute and linked to researchers at Lancaster University and also in Brazil. So a couple of years back, Chris developed a technique for immune marking culicoides, where you basically spray the environment with a food-based protein, and then from a catch you see if you can detect these markers in the insects themselves. So Chris and the individuals at Lancaster and Brazil are looking if they can move this technique from culicoides into sandflies. And not only will this give data on resting sites and breeding habitats, but it will also able to look at dispersal in the field. And all of this dispersal study is going to be carried out in Brazil, apart from some of the initial validation of the technique. So that was our first call, but at the end of last year and the beginning of this year, we introduced our second call, which is called Transformative Science. So this one, we were looking for much more novel ideas, much more blue sky science, and we're actually open to projects that had a bit of a degree of risk to them. We funded a total of 2 projects from this, and they both started either last month or this month, so they're very new. So our first project is being run at the Natural History Museum by Dr. Martin Hall. And Martin has basically been looking over the past few years at micro-CT visualization techniques. So what he does is he takes a specimen, he takes 3,000 X-ray images from around the specimen, and then uses computer software in order to reconstruct the image. So you can kind of look at the outside, you can take away layers, look at the inside, and he's used this to look at the parasitization of ants. However, Martin is wanting to now move this to look at something a little bit smaller, so he's going to look at sandflies and blackflies and the parasitization of those. But basically he's got 2 hypotheses for this. Firstly, he wants to look how leishmania actually deforms the sandfly midgut. So it's believed that once a leishmania parasite enters the sandfly midguts, it's able to transform the stomadeal valve in order to move around throughout the sandfly, and also compare the midguts of different blackflies. So different blackflies are able to have different levels of parasitization, and his idea is that Onchocerca is able to develop in the midguts of some better than others. So he wants to compare the different midguts to see actually how they look and see if that's the reason for the difference. But this will link to researchers in Bangladesh and Ghana. And our final project that we funded in this call, and the final one we've got going at the minute, is led by the University of Warwick, and it's looking at understanding host vector parasite contact rates. So this is going to look at 2 different vectors, sandflies, which transmit leishmania, which causes leishmaniasis, and blackflies, Onchocerca, which causes river blindness. So what the researchers are going to do here is compare 2 types of area, one of high disease prevalence, high biting rate, one of low disease prevalence, low biting rates, and look at the heterogeneity in exposure to salivary proteins, just to see if there's any difference between the 2 areas. So he's going to do this through anti-vector saliva ELISAs. One already exists for sandflies, but they're going to create a novel one for blackflies. And the idea is that eventually this data will feed into transmission models to show where control is best suited in order to target the diseases. And this is going to link to researchers in Sudan and Ghana. However, very excitingly, we have decided this year that we're going to run a 3rd and final call, and this time we're really mixing things up a bit and doing it very differently. So we started by getting our expert management board, which are 8 international experts around the world, to come up with 6 ideas that they thought needed to be addressed within the biting midge, sandfly and blackfly communities. And these ideas have now gone actually onto our website, where our Gnatwork members are voting for the ideas that they think will benefit the community most. So this is our vote at the end of the first week of voting. So it's still quite close, so there's still room for movement in there. But the idea is once the ideas are voted for, our top 3 will go onto our website, where people can basically tender to do the research. And they'll be selected based on experience and CV level. But our voting is still open. It's got 1 more week. So if you want a say in what projects we fund next, basically head onto our website. You can sign up as a member for free and read our projects and get voting. So finally, how can you get involved? So anyone can join the Gnatwork. It's completely free to sign up. And we currently have 260 members and counting from 53 different countries. Also, we share entomology news, upcoming entomology conferences through monthly newsletters and social media. And over the past couple of months, we've really looked at getting a lot more resources onto our website. So we now have a number of freely downloadable protocols, anything from molecular barcoding, truck trapping, DNA extraction, which are all freely available and you can just download straight from our website. And please do get online and vote if you want to say in what projects we do fund. So with that, I would like to just thank our network management team, so Simon, Mary and Alex, who is our network administrator, who started about 9 months ago, and our panel of international experts, which are experts on blackflies, sandflies and biting midges from around the world. So thank you very much. Take any questions.